right, good morning, everyone. It's my great pleasure to introduce our speaker of the ADRC series today, Dr. Tim Huang, who is an assistant professor at the Stanford Brennan Priebus Institute. Tim did both of his undergrad and PhD study in Canada, and first at University of Alberta and then at University of Calgary, respectively. And then he came to the States and did his postdoctoral training first at Scripps and then at Brennan, where he stayed on to become a faculty member. So I'm pretty sure many of us have seen him around for a while. That's how I got to know him in the community, and we became friends. So Tim has been studying the molecular mechanism of Alzheimer's and has really spearheaded many seminal discoveries, and specifically his main dish, I want to say, of work has been focused on this protein SORLA, which is a trafficking receptor whose polymorphism has been associated with AD risk. Tim has really gone very deep into elucidate the protective role of SORLA, both in neurons and in microglia. And I also been followed closely with his recent work, which shed light on the role of TREM2 in regulating tau propagation in microglia. So I found that's a very comprehensive and impressive piece of work. So I'm so glad to have him come over to speak with us today. And without further ado, let's welcome Tim.
Thank you so much, Shu. Always a pleasure to speak with friends here at UCSD ADRC. So I'm here to share with you some recent work on SORLA and tau, and also to give you guys a recap on some of the work I've been doing with the lab on SORLA for the last 10 years. So this is what we're going to talk about today. So basically, we'll go through some of my earlier postdoctoral work on SORLA regeneration and A beta generation and uptake. And then we'll go to some unpublished work characterizing the effects of SORLA upregulation on tau pathology and pathogenesis. So that's our menu for today. Okay, so this crowd does not need this introduction, but I'll go through this very quickly. So Alzheimer's, it's a very serious disease. The symptoms, they include memory loss, spatial disorientation, confusion. Basically, individuals, they gradually lose their everyday function. They're not going to be able to function by themselves at the end. So early onset AD is generally classified as AD onset before age 60 or 65, and late onset, which is the most common form of Alzheimer's, is above age 65. And so when we talk about Alzheimer's disease, we always bring up August Dieter, which is the first diagnosed patient for Alzheimer's. So at age around 50 to 55, she began to complain of memory problems. And she was diagnosed by Alzheimer's in Germany. And basically, he noted two distinctive pathologies in her brain. So these are the tau tangles and beta amyloid plaques. And so a few tidbits about Alzheimer's. So 1 in 3 seniors die from Alzheimer's or alternate dementia. Currently, 6 million people suffer from Alzheimer's in America. This is projected to increase to 13 million by 2050. And currently, the estimated cost for AD care is about 345 billion. This is projected to increase to the trillions by 2050. So this is going to be a serious problem in the future. So Alzheimer's is part of a group of dementias. It is actually the most common. So about 70 to 80% of dementias are Alzheimer's disease. There's other types: vascular dementia, Lewy body, frontal temporal dementia. Alzheimer's, however, is primarily characterized by the appearance of A beta plaques and tau tangles. So it is primarily defined by the A beta plaques. So it is a secondary tauopathy, meaning that tau also appears. It's an important pathology, but both of these pathologies are occurring in Alzheimer's disease. So tau also belongs to a group of primary tauopathies. These include frontal temporal dementia, Pick's disease, cortical basal degeneration, PSP. So these are all primarily defined by the appearance of tau after onset and post-mortem. So I just want to talk about A beta and tau a little bit. So these A beta plaques, they're enriched for these peptides that are about 40 to 42 long that aggregate outside of the cells. Whereas tau, they form these long fibrils inside of neurons in its hyperphosphorylated form. So during progression of AD, before individuals have any problems with memory, about a decade or more, they begin to build up A beta, both in the brain and the CSF. And other events include microglia and astrocyte activation and buildup of tau pathology during mild cognitive impairment. So when individuals start to see memory problems, and then at the late stages, we see changes within brain atrophy and other features of pathology. So where does A beta come from? So A beta comes from the processing of a type 1 transmembrane protein called the amyloid precursor protein, and it gets cut twice. So it gets cut at a more N-terminal region right here by beta secretases, base 1 and base 2. It also gets cut at close to the membrane, so right in the membrane by the gamma secretases. And it makes this 40 to 42 peptide, which is prone to aggregation. So there are familial mutants that will basically accelerate the generation of this 40 to 42 peptide. These include commonly the Swedish mutation, London and Indiana mutations, which are all familial AD mutational variants. So tau is normally bound to microtubules. So it's a microtubule stabilizing protein. And upon hyperphosphorylation, it gets dissociated from microtubules and is prone to aggregation. Okay, so I always bring up this figure that shows the genetic landscape of Alzheimer's disease. So this is kind of a more or less complete portrayal of all the risk factors that are associated with AD. So there's a lot more, but I like this figure by Celeste Koch and Alison Grote because it captures really what's happening within these genetic variants. So we can see up top here, PS1, PS2 APP, so this is the gamma secretases and the type 1 precursor, membrane precursor. These are all involved in enhanced A beta generation. So these mutations, when people have familial AD mutations, basically there's a 100% chance almost that they will have AD before age 60. So usually they manifest problems between age 40 and 50. And so these are very rare mutations. So on this x-axis, you can see basically this is the prevalence of the different variants along the x-axis here. And so basically, they're very rare, but they're fully penetrant. So these sporadic variants, they don't 100% manifest Alzheimer's. So basically, they'll increase your risk, but their penetrance is not at 100%. So the highest risk factor is this cholesterol lipid carrier, ApoE. And the E4 variant is associated with high risk. E3, more people have E3. This is kind of the common variant, which is more or less neutral. And we have an E2 variant that's of reduced risk. As you can see, there's also other variants, including the Ig type receptor in microglia, TREM2, that's responsible for activating microglia in response to immunogenic stimuli, as well as SORLA, which we're going to talk about today. So these are kind of, the focus of the current lab is looking at signal transduction trafficking within different cell types. So TREM2 is expressed in microglia, SORLA is expressed in a lot of different cell types. In humans, it's mostly expressed in microglia, but we'll also talk about a neuronal function for SORLA. So these sporadic variants basically comprise the late onset form of Alzheimer's, which comprises over 90% of Alzheimer's disease. So what's interesting is I took away all the factors that are not transmembrane or extracellular. So, a lot of these AD risk factors are either transmembrane components or they're extracellular factors. And just one more tidbit. So this is a study from Richard Mayu. And in some populations, he shows that SORLA is actually enriched beyond what is expected for an AD population. So in this particular publication, he shows that there's a 50-fold enrichment beyond what we normally would predict that would be for a AD risk factor. Okay, so this is what we're going to talk about. First of all, just maybe the last decade of work looking at some earlier research on SORLA regeneration A beta and AD from other groups as well as my own lab. So what is SORLA? Why do we care about it? So it's a type 1 transmembrane trafficking component. So it has a VPS10 trafficking domain, similar to other trafficking components, for example, sortilin, as well as the SORCS 1 through 3 family members. And it also comprises LDLR repeats, which in LDLR or LRP1, this is the ApoE, proposed ApoE binding region for these trafficking components. So in the old days, it also has other domains, for example, beta propeller, which is also called the YWTD EGF-like repeat, and also fibronectin 3 repeats here, and a unique cytosolic tail. So in the old days, before genomics, this was called a mosaic receptor. It has a lot of different domains. It doesn't really have any similarity to other receptors. So before anybody linked SORLA to AD, SORLA became on the radar for people that study this organism. So this is a freshwater hydra species. It basically likes to prey on other smaller freshwater organisms like Daphnia. And the reason why people were interested in it is because, first of all, it doesn't seem to age. So it doesn't seem to, this organism seems to be able to replicate perpetually. And secondly, it is able to regenerate its tentacles. So a group from Germany, Schaller, they identified an 11-mer peptide called head activator that stimulated the regeneration of its tentacles. And they were looking for targets that might affect this HA peptide. And so basically through some biochemistry, they cross-linked HA to putative binding proteins. One of them, basically they pulled out the Hydra ortholog of SORLA. So and they called it head activator binding protein in the earlier days. So this is kind of, where this story ended. So we don't know anything much more about HA and SORLA biology, except that some follow-up work from the Schaller group, they showed that SORLA is probably expressed in regions that might be associated with active growth in this hydra. So other groups that also showed that SORLA, or LR11, which is an earlier name for SORLA, is also associated with, for example, atherosclerosis, so specific medial to intimate transition of smooth muscle cells during atherosclerosis. And so we began thinking in the lab, well, SORLA can mediate outgrowth of stuff like tentacles and hydra, and also, has motogenic function in smooth muscle cells. And so a project by Jessica Stuback, which was a graduate student in the lab, we began to wonder with her, can SORLA mediate processes in the brain that are associated with some type of outgrowth? So we were lucky to have a transgenic mouse that overexpresses SORLA. We'll talk a little bit more about that later. But what Jessica did was just compare the length of neurites in DIV3 mouse neurons, either wild type or neuron that's overexpressing SORLA. She noticed that in the SORLA TG neurons, the neurites seem to be a little bit longer. She also took 2D neuron cultures. She injured them and basically looked at the neurite outgrowth, compared them between the SORLA and TG neurons. And she found that in the SORLA TG neurons, the neurites tended to grow out into the injured region more quickly. So SORLA is a type 1 transmembrane protein. It gets cut by alpha secretases to release a soluble SORLA form. And in these SORLA transgenic mice, we see more of this soluble SORLA being shed out into the median compared to the wild type neurons. And so one possibility is that the neurites might be growing faster because there's more of the soluble SORLA flowing around in the media. So Jessica purified soluble SORLA, and she applied it to these injured neurite cultures, and she found that the increasing amounts of SORLA can also enhance the neurite outgrowth during injury. So she also explored the mechanism. So SORLA, it's shed, it's floating around in the media. Basically, what this does is this activates the EGFR MAP kinase pathway. So she took various inhibitors to inhibit this pathway, and she finds that if you add SORLA, adds these inhibitors, you don't get this neurite outgrowth enhancement. So yeah, so about 20 years ago, this is when SORLA became kind of prominent on the AD radar. So people in Alzheimer's became interested in SORLA. So one of earlier studies comes from James Law at Emory. So they were looking for blood biomarker for Alzheimer's disease. And they found that in patient lymphoblasts, the SORLA levels, or SORLA is also LR11, it was decreased in AD patient lymphoblasts. So later on, Peter Hislop and other groups, they've identified numerous single nucleotide variants that are associated with Alzheimer's disease. And so this is a figure I made in 2017. So there's a lot of different coding mutations that are associated with SORLA. And I said to myself, I would update this every year, but I kind of gave up. So these SORLA mutation variants, they occur in different domains within the SORLA open reading frame. So Alzheimer's, they did a much better job, and their picture is much prettier, so I'll stick with this. So there's, as you can see, there's a lot of frame shift, missense, also some silent mutations that are occurring in various domains within the SORLA protein, even within the cytosolic trafficking domain. So clearly there's some link between SORLA and its function, perhaps trafficking, perhaps other functions that will impact cellular function that's associated with Alzheimer's disease. So just to, some earlier work showed that SORLA is a trafficking component for APP. So APP is that type 1 transmembrane component that gets cut into A beta. So SORLA is comprised of these different domains, and it interacts with APP through LDLR-like repeats. So APP, when it comes from the membrane, it gets sorted into the endosome. From the endosome, it gets sorted to other places in the cell. And when it gets sorted into late endosomes, it gets cut by base 1. So this is the rate-limiting step for A beta generation. So some earlier work from Olaf Anderson showed that SORLA binds to the retromer complex at the early endosomes, and this helps shunt it from the endosome to the Golgi, and this bypasses the trafficking to the late endosomes. So some of our work, we show that when SORLA binds at the sorting exon 27 subunit within the retromer complex, this has a canonical function for trafficking APP back to the membrane. And what this does is basically this promotes alpha secretase cleavage, which is right here in the middle of this A beta domain. So basically it can preclude the processing of A beta when we have this alpha secretase cleavage event. So some work from Tom Wilnau's lab is very interesting. Basically, he showed that SORLA can bind A beta directly and traffic it to the lysosome for degradation. So he shows this in cultured cells. So basically, A beta can bind the VPS10 domain, and this results in trafficking of A beta to the lysosome. So in the same study, he generated SORLA TG mouse. So this is human SORLA that's expressed from the CAG promoter at the ROSA 26 locus. So this is global overexpression of SORLA. And what he shows is that SORLA overexpression can decrease A beta levels. Okay, so he shows that trafficking is, SORLA can traffic A beta to the lysosome in cultured cells. So we wanted to test whether this is the same case also in microglia. So Bing and Tofing in the lab, they took embryonic stem cells and they made stop gain, the AD-associated stop gain mutations for SORLA, compared that to the TREM2 AD-associated R47H variant. They differentiate these ESCs to microglia. They did some experiments in vitro. I just want to point out they did a very interesting experiment in vivo. So basically, they fully differentiated this microglia and it injected it into TGCR-ND8 AD brain. And then they inserted a microdialysis filter and looked at the clearance of A beta in these microglia injected brains. And what they see is that the wild-type microglia, they can clear A beta for some period of time, up to 18 hours, whereas these microglia that have R47H or a 744X stop gain mutation, they have troubles clearing A beta. So there's so they also have some experiments in vivo. In vitro. I welcome you guys to check it out. So this is all in all saying that when SORLA its function is impaired, that when SORLA function is impaired, that basically we can also disrupt the A beta uptake and trafficking in microglia. Okay, so next question that we wanted to ask is whether or not SORLA has any downstream effect of A beta. So this is to say that if we have increased SORLA levels, we know that A beta is toxic. Is there some type of protective effect to resist neurons to A beta synaptotoxicity? And so we were very fortunate to get these SORLA TG mice from Tom Wilnow at Max Delbruck. And basically, this is the question that we want to ask. So if we pulse these mice with A beta, is there any type of protective effect in these SORLA TG mice that are overexpressing SORLA? And so these are stereotactic injections that we made in wild-type SORLA TG mice. And basically, we tested their memory behavior using the Morris water maze test. So we trained mice to find this platform using these fiduciary markings. And then after 5 days of training, we see whether or not these mice can find the same spot. So wild-type mice, when they're injected with A beta, they don't do very well. So basically, they're kind of swimming around. They can't find the platform. And however, these SORLA TG mice, they kind of are a lot better. So basically, when we inject them with A beta, they're almost similar to wild type. So they're able to learn almost normally. They're able to find a platform. So this is basically a probe test looking at the mice crossing this region. So we let the mice recover for a week. We let it go through Morris water maze. After we're finished with the behavioral test, we looked at the A beta levels. So with this paradigm, this acute injection, we don't see any difference in the A beta levels between the wild type and SORLA TG animals. So we're looking for a mechanism. So there's a lot of A beta receptors out there that have been characterized. And one hypothesis that we came up with is that maybe SORLA, because it's on a cell surface, it might affect other A beta receptors. And so I was going to go through the entire box of our freezers, A through Z, go through basically do IPs for all the different A beta receptors. Luckily only had to stop at E. So we got an antibody from Elena Pasquale at SBP, and we found that EPHA4 could interact with SORLA. So what is EPHA4? So it's a receptor tyrosine kinase that binds ephrin ligands. So this is important during the development of early brain. So basically, it's involved in providing axon and reacting to axon guidance cues. And this is required for proper neuronal wiring during development of brain. So basically, ephrins will meet ephrin ligands will meet ephrin kinases. This results in superclustering of these ephrin kinases, hyperphosphorylation at the C terminus, and this has downstream events that involving the Rho GTPase pathway that we get growth cone collapse. So this is developmental. So some more recent work shows that this EPHA4 is also an A beta receptor. This causes a similar effect. So A beta causes clustering of EPHA4 at postsynaptic densities. This is synaptotoxic. And basically what happens is A beta can, through EPHA4 activation and clustering at postsynaptic densities also impair synaptic function. So because SORLA could interact with EPH4A4, we just wanted to see whether or not SORLA can affect these two different phenomena. So this is just a figure shown here that this ephrin ligand, it can induce clustering EPHA4 in wild-type neurons, but not so much in TG neurons. And we have a bunch of experiments showing that SORLA can also suppress EPHA4 activation clustering in neurons and reactivity to A beta. But I just want to show this figure here that we have a model. So basically, SORLA will bind EPHA4. It prevents the clustering of EPHA4. And basically, this is probably a synapto protective event. So these are a cohort of about 50, 80 patients. And what we see is when these are IPs, and what we do is IP for EPHA4 SORLA that we plot for either total EPHA4 or the phosphoactive form. And what we see is that if there's more interaction between SORLA and EPHA4, we see less phosphorylation and activation of EPHA4. Okay, so just in summary of this section, so there's a lot of data out there that shows that SORLA can reduce A beta generation. So some really neat results from Olaf Anderson's group shows that he made a haploinsufficient mini pig, and he shows that a lot of the CSF patient profiles can be reiterated by haploinsufficiency of SORLA, haploinsufficiency of SORLA. So in different cell models, we show that, SORLA can traffic APP to bypass base cleavage at the endosome. There's also some really neat data from iPSC models out there for SORLA deletion that shows endosomal enlargement, for example. So from our lab, we showed that SORLA can confer resistance to A beta proteotoxicity and enhance neurite generation. And so we also see that SORLA can mediate A beta uptake. So this is where we're at. So we know that SORLA can mediate resistance to A beta toxicity, has a function in suppressing generation, and also mediates clearance to some extent. So the question we want to ask is virtually nothing is known with respect to what SORLA can do to tau. So can it affect tau phosphorylation? Does this affect glia activation, all these downstream events? So this is the big question we want to ask. Can SORLA upregulation confer resistance to tau toxicity? So this works spearheaded by Huang, which is sitting here in the second row here, as well as members of the SORLA tau team. Okay, so we took Virginia Lee's PS19 model. So this is a P301S tau, which is a familial mutation from a Dutch family that's basically expressed from the prion promoter. So this is 1N4R tau. And we crossed it into SORLA transgenic animals. And Huijie looked at phosphorylation of tau at 3 months and 9 months. And what she observed is that at 3 months, we don't see much difference in the phospho-tau levels. And there's, maybe a trending decrease in the SORLA TG animals crossed with the PS19. However, at 9 months of age, she really does see, tau phosphorylation, a little bit more consistency with a little bit more consistency in the PS19 animals. This is suppressed in the SORLA TG PS19 animals. So what's really neat is that every time we see tau phosphorylation, we also see an increase in astrogliosis, or at least upregulation of GFAP. This is very tight correlation. So basically, if there's a increase in phospho-tau levels, so this is the 88 epitope, the 202, 205 phosphoepitopes, we also get increased in GFAP levels. So this is some staining. So basically, PS19 compared to SORLA overexpression PS19 seems that SORLA TG PS19 seems to be suppressed in terms of the phospho-tau levels. So this mouse model at later ages is also associated with astrogliosis. So there's some GFAP staining in 6-month-old PS19 mice. And so Hui also did some staining in our older mice, and basically she saw that PS19 has some pretty prominent astrogliosis, whereas SORLA overexpression can largely attenuate the astrocyte astrogliosis morphology. And she also sees the same thing with microglia activation. So CD68 staining is pretty upregulated in the PS19, is suppressed with the SORLA overexpression. So this is a phenotype that's kind of new to me. So basically, Virginia Lee, she sees this ventricle dilation. And so we wanted to see whether or not SORLA had any effects on ventricle dilation in these coronal sections. And what we see is, although, this ventricle dilation phenotype is a little bit variable, we see that the SORLA upregulation can suppress the ventricle dilation to a good extent. So next thing we wanted to do is just to take a look at the proteomic profiles in the hippocampus of aged mice, so wild-type SORLA TG, also the PS19 or SORLA the PS19 combined with the SORLA TG. And you can find that, the proteomics profiles separate between these different genotypes. And what's really interesting is that we see some DPs in PS19 versus wild type that we don't see in the TG PS19. So we're losing some effects that are seen with PS19, the PS19 pathological protein landscape. So doing a gene ontology analysis of these components, a lot of them are involved in something to do with the synapse, either synapse glutamatergic synapse or postsynaptic density, for example. So if we look at the profiles of all these synaptic components, we see kind of two patterns. So a large group of them are downregulated PS19. They're kind of a little bit more upregulated TG PS19. But what I wanted to point out is we have this really interesting group of these synapse-related proteins that are upregulated PS19. They're kind of more or less downregulated when we cross into SORLA. So I just want to introduce to you guys a little bit about ApoE and the C1q components in this cluster. Okay, so C1q is super interesting. I'm sorry, so interesting. Beth Stevens, she has some earlier work that shows that C1q, it's part of the complement cascade. And it's required for eliminating weak synapses, either in the retinal geniculate or in the visual thalamus. So basically what happens is astrocytes, they secrete TGF-beta, which then induces neurons to secrete C1q, which then starts to cascade to generate these opsonins, that C3b, for example, that tags weak synapses for elimination by the complement receptor 3. So this is earlier during development. So she has some really interesting work with Huang later that shows that A-beta can actually induce C1q in aged animals. So this is APP animals. And this also tags synapses for pathological elimination. So this is something like a synaptic stripping during A-beta pathology. So we were super interested that this was upregulated in PS19 and not so much in the TG PS19. So some work from Ben Barres also shows that the ApoE variants are also associated with C1q levels. So these are some humanized ApoE mice, and she shows that if the mice are expressing the high-risk AD variant, there's higher C1q levels in the hippocampus, whereas the ApoE2 humanized mouse, there's not so much C1q in the hippocampus. So just in relation to this ventricle dilation phenotype, some work from Dave Holtzman's labs also shows that if you delete ApoE, a lot of this ventricle dilation goes away. This is the same thing for C1q deletions. This is some work from Jesse Hansen's lab at Genentech, and he also shows that C1q deletion can also attenuate the ventricle dilation phenotype. So it's really interesting that when we upregulate SORLA, we can also suppress the expression of these two potential upregulators of tau pathology. So Hui, did some validation. So she looked at ApoE levels. It seemed to be upregulated, PS19, downregulated when we cross it with the SORLA TG. She also sees induction of C1q in the hippocampus in the PS19. This is downregulated with this SORLA upregulation. And so basically, we also saw markers of microglia activation. So when microglia are activated, when they start to strip synapses, they begin to take up synapses. So Huijie did some MRS analysis looking at C1q and PSD95 puncta in PS19. And she shows that there's quite a bit of puncta that are taken up in microglia and PS19 hippocampus, not so much in the SORLA TG PS19 hippocampus. So Jay Bhatnagar, who was a high school student, he did some electrophysiology with us with Juan Pina Crespo. So basically, they're looking at LTP response in the dentate gyrus. So we see some impairment in LTP response in the PS19 animals, and this is largely suppressed in the PS19 animals overexpressing SORLA. So Huijie also did some single nucleus RNA-seq analysis comparing wild type PS19 and SORLA TG PS19 to look at whether or not these pathogenic profiles in certain cell types are also reversed in with SORLA overexpression. Just looking at microglia profiles first. So there's different states of microglia. Probably the most famous is they're usually in a homeostatic state. When they're in a diseased state, they become activated. And in 5XFAD models, Ido Amit, for example, he showed that there's various distinctive profiles that give you disease-associated microglia or DAMs. Okay, so we can identify DAM and homeostatic microglia in our single nucleus RNA-seq data set. But what's very striking is that these homeostatic components are downregulated in the PS19 hippocampus, so PS19 versus wild type, but they're upregulated in the TG PS19 versus PS19. So it's upregulated the SORLA TGs in the PS19 background. So conversely, with the DAM genes, so the PS19s, they upregulate the DAM genes, and the TG PS19, they downregulate the DAM genes in these microglia. So I'm just highlighting C1q, ApoE, showing that potentially what we're picking up with the proteomics, downregulation of C1q, C1qa in the TGPS19, as well as downregulation of ApoE, could be due to what's going on with the microglia induction of C1q and ApoE in pathology. Okay, so we looked at all different cell types using transcriptomics, and I'm just showing you astrocytes here. So astrocytes also show a distinctive disease-associated profile. And similar to microglia, we see upregulation of disease-associated genes in astrocytes in PS19. This is downregulated in the SORLA overexpression in the PS19 background. So it can also reverse disease-associated signatures in astrocytes. So one question that we want to ask is SORLA regulating something that is kind of controlling the glial state? That we're looking for some type of upstream regulator for glia activation. And so Christina, she helped us with some bioinformatics looking at cell phone DB databases. So we're looking for the cell-cell interaction pairs that are enriched either in PS19 that don't occur in wild type or TG-PS9, or it's vice versa. What we found interesting is that she identified this plexin B pair with SEMA4D that occurs in PS19 animals only. And so the reason why we find this interesting is that SEMA4D and plexin B, they're a ligand receptor pair that's also similar to EPHA4. It's also involved in growth cone collapse and axon pathfinding during development. So neurons will express plexin B, meets SEMA4D, for example, and it will mediates growth cone collapse to properly wire neurons to the right place during development. And the reason why we were excited about this is because Huijie noted that a lot of disease models, either multiple sclerosis, HD-AD, human AD brain, The semaphore-D or plexin B pathway is upregulated. So what's really interesting is that they have semaphore-D targeting antibodies that are currently in, so this is pepinmab. So they have these antibodies targeting semaphore-D right now in clinical trials for HD. And I think they just started the AD trials in early phases for this semaphore-D inhibiting antibody. So this is some really interesting work from Roland Friedl's lab. So what he shows is that normally plexin B semaphore-D signaling keeps glia away from plaques and the glia are activated. But when he knocks out plexin B1, basically the glia are able to infiltrate and latch onto the plaque, but they become inactivated. So there's some type of some type of distancing event, some pathological distancing event in these glial nets that's mediated by the plexin B1 and semaphore-D activation. And so this is really interesting. Once again, we come to this interaction pair, receptor ligand interaction pair that's involved in development, but it also comes back again as upregulated during pathology. So this has never been shown to have anything to do with tau. So we wanted to ask the question, well, is this pathway upregulated in response to tau, and is this affected by SORLA? So basically, Huijie showed that plexin B1 is upregulated PS19, and it's downregulated when we overexpress SORLA. Same thing with plexin B2. So it's highly enriched in microglia. It's upregulated PS19. It's largely downregulated when we overexpress SORLA. So we were thinking, now we have a new unique target for tau. So basically, tau can induce plexin B1 semaphore-D signaling. This might be pathogenic. Can we downregulate this and show that it might be protective? And we were thinking of different strategies, maybe shRNA. But we got lucky. So basically, there's this group that shows that so semaphore-D and plexin B signaling is also involved in osteoblast differentiation and maturation. And this is specifically important in osteoporosis. So basically what we want to do is we want to inhibit semaphore-D plexin B1 signaling, and that will enhance osteoblast function, and this will restore osteoporosis. So Hiroaki Suga's group, they identified through library screening this cyclic peptide that is able to inhibit semaphore-D signaling with plexin B1. And they show that, so basically they overoptimize mice, and they show some bone degradation. But when they treat these mice, overactualized mice with this peptide, they can restore a lot of this osteoblast function. So I'm sorry to leave you guys with a cliffhanger here, but Huijie injected these mice a week and a half ago. So we're really excited to see what was going to happen with the PS19 mice when we inject them with this peptide. So hopefully more information in the near future. So just a summary, this is what happens. So basically, we think that SORLA is going to be protective. It has some effect in reducing phospho-tau levels, but also other things. So it suppresses expression of upstream mediators tau, for example, C1q, ApoE. It also suppresses synaptic impairment. It reverses pathological transcriptomic readouts in pretty much all the cell types that we see. And it reduces glial activation, or so we hope, potentially through upregulation of plexin B and semaphore-D. So it does a lot of things. During when we were working on this project, a really interesting paper came out from Dudley Strickland's group at UMaryland. Basically, these guys show that tau can also bind directly to SORLA at different pHs, so acidic and neutral pHs, and basically it mediates tau internalization in CD. So we don't know how this fits into our story, because their work in reporter cell lines, so the tau FRET reporters, indicates that SORLA is required for internalization and pathogenesis, genesis, whereas we showed that SORLA upregulation is probably protective. So we're going to take a little time to try to figure this out. So how does SORLA being a tau receptor fit into what we see? What's really interesting is that we come back to this kind of reframe where SORLA is suppressing pathways that are important early in development that come back and upregulate, that's upregulated or active in pathology. So for example, EPHA4, which is involved in growth cone collapse, or C1q, which is involved in eliminating weak synapses, or plexin B semaphore 4D signaling, which is also involved in growth cone collapse. So these pathways, they come back again during pathology, but SORLA suppresses them. So this is kind of like a really neat reframe that we keep on seeing in our studies. So I want to take a couple more minutes to give some final thoughts. So we're all different. So we all react differently to stress. So if we take age as a stress, we're all going to react differently to age. So some individuals, for example, August Dieter, she'll manifest, for example, early onset dementia. Some other individuals, for example, Tom Parr, who is probably one of the oldest documented human beings that we see, basically, he had a very healthy brain and in postmortem analysis, and it didn't seem like he had a very healthy lifestyle. So this is saying that there's a huge spectrum of how we react to disease and stress. So I just wanted to tell you guys a story about another source of stress, for example, ricin. So ricin's a poison, so it's a ribosome inactivating protein from castor bean. And in the 80s, Soviet defector Georgy Markov, he got stabbed with a Bulgarian umbrella. So this delivered a ricin pellet into his leg. He had fever symptoms, he had vomiting, he had really terrible, symptoms. He had tough time breathing, and he passed away 3 days later. So the same assassination was attempted on Vladimir Kostov. He was another Bulgarian defector. He survived the attack, and we're not quite sure why. So there's numerous. So they found a pellet within him, but he was wearing a thick sweater that day. So there's one theory that maybe the penetration of the ricin was not complete and he got less poison. So he did get symptoms for a few days, but he fully recovered. He also wrote a book called The Bulgarian Umbrella. So this is to say that ricin is very toxic. So it inhibits a protein synthesis at very low concentrations. This causes death. But there's some factors, maybe Kostov's sweater, that prevented this event. So basically, he was able to survive. And so with respect to AD, we have proteotoxic stressors. We have A beta, we have tau. These are associated with events such as glial and neuronal dysfunction. And eventually, this causes the synaptic dysfunction and cognitive impairment and basically wasting away of the brain. So there's currently, FDA-approved antibodies that are pretty good at clearing A beta, but they don't have a huge effect on, rescuing cognition. But what's really interesting is that there are a lot of individuals in the clinic that will develop AD pathology. So pathology comes at least a decade before any onset of these symptoms. But some individuals, they will have it, A beta or tau, even tau pathology that their entire lives, they don't show very much of a problem. So they have some protective factors, and we're starting to build a list of these factors, what they might be and how they might be protective. Some of the more interesting ones are for example, ApoE Christchurch or Relin mutations that we find that are able to bypass the complete penetrance of the PS1 familial AD mutations. And hopefully, with more work, we're able to add SORLA to this list. And once we know more about how SORLA functions, how it mediates neuroprotective effects, then maybe we can try to devise strategies to utilize these pathways. Okay, so there's a lot of people to thank. So first of all, I'd like to thank my previous postdoctoral mentor, Washi Shu, who unfortunately passed away 3 years ago. Without this, none of this SORLA work would have come to pass. I also want to thank members of the soluble SORLA neurite outgrowth team, Jessica Stupak. We also had some really good help from Lena Pasquale at SBP. The SORLA Tau team, spearheaded by Huang, who's over here. She'll take autographs after the seminar, as well as the SORLA Tau team and the rest of the lab. Special thanks to Kevin Ip at SBP for bioinformatic analysis with Christina Shi, proteomic analysis from Alex and Svetlana, and bioinformatic analysis from Rabbi. We're very grateful to Tom Wilnow for providing us with this mice. So if we didn't get the mice, we wouldn't be able to do any of this work. So, and yeah, this is Weijie here, and she's